a different thematic. In this case, C3C around the islands. I will be the chair, and I am pleasure to present the keynote speaker today. That is Professor Brent Emerson. Professor Brent Emerson started his career indeed studying phylogeography with using as models some insects and in Canary Islands with very interesting studies about phylogeography of Canary Islands animals. Then at same point he was based in UK, but at some point he moved completely and full minded to Canary Islands. He started a new new strategy in his career, and is now a leading researcher in Macaronesian evolutionary studies, using again arthropods and insects in particular as model systems, but also working in theoretical topics in molecular biology, molecular ecology. And as you will see today, he will present very novel research he's doing with studies using soil organisms as models, Colembula as and other organisms, and also, of course, keeping working with insects as models due to the high diversity in Canary Islands. That is a fantastic place to study evolution, as you will see. I give the floor now to Professor Brent Emerson, hoping that we all enjoy all his fantastic research in Canary Islands.
Okay, thanks very much, Paulo. I'll just start by saying a lot of what I'm going to present today originates or has been stimulated by Paulo and conversations we probably had about 12 years ago to design a sampling framework in common between us. And so pretty much all the data here derives from those early conversations in many ways. So what I'm going to talk about today is DNA-based approaches for landscape scale understanding of arthropod communities. And why is this not letting me check? Okay, let's see. Okay, sorry. Yes, there we go. It's changing. Great. So one of the big problems we're confronted with when we want to do community scale analyses of arthropods is just the logistics. There's a lot of individuals in a community from a lot of different species, and unless you're an expert, you're not going to be able to identify them very well. So this is one of the big issues we've had to first overcome. And that's where the conversations with Paulo came in, and we basically extended the COBRA protocol developed by Pedro Cardoso and Paulo and others to include a molecular component. And so that enabled us to sample massively, identify individuals to what we call parataxonomic units, and then incorporate a molecular step to validate those species or to refine further our understanding. And that's worked very well. It reduces the overall taxonomic load, gives us great comparability across different studies through the DNA sequences, and additionally reveals cryptic diversity, either population structuring within species or new species themselves. But most importantly, this data also gives us some information about individual species dispersal limitations. So I'm just going to sort of briefly show you how that works here. So here's just an example. It's not hypothetical. These are 10 plots we've been working with. So in each of these plots, we might, across these plots, sample 20,000 individuals. And for each species or parataxonomic unit, we'll sample 4 individuals and generate mitochondrial sequence data. And that can give us instantly fundamental information on dispersal ability. So, for example, in the case on the left here, species 1, all variation is everywhere. That tells us that this is a panmictic species, no dispersal limitation over the evolutionary timescale of the sequence variation. Whereas on the right here, we see lots of structure. That instantly tells us, yes, something's going on. There is dispersal limitation going on among some sites. And because we're using sequence data, we can further quantify that in terms of a phylogeny and even look at, if you like, the timing and the relatedness among major areas of dispersal limitation. So that for us is like, it's incredibly useful information as I'll hope to show you as we proceed. So what I want to present here is work from the first 10 sites where we implemented this protocol. And this is from a project working together with Paula. And so we've got 10 sites across the Yanaga Peninsula in the cloud forest along the dorsal ridge of that peninsula. So there's 50 by 50 meter plots. We apply basically a slight modification of the Cobra protocol, and that's about 27 hours of sampling with a range of different sampling techniques. Here we focused on beetles and spiders, and this data alone gives us information about presence and absence and local abundance and even microhabitat information. Now, what I'm going to present to you is in the more specific details can be found in this paper here for anyone that's interested. Okay, so I'm kind of going to go quite lightly. So across these 10 plots, we're just going to look at the beetles. That's the group that we've placed most focus on until now. Of course, the spiders are next, but we need more hands basically. But the beetle data turned out to be incredibly interesting. So we sampled just over 23,000 individuals and identified 154 what we call presumed biological species based on the molecular data. There are more species for which we couldn't get sequences. So there's probably really about 180, but these 154 gave us good sequence data. 146 are native or endemics. So it's all of a sudden immediately interesting. There's very little evidence for the cloud forest habitat of the canaries being compromised by introduced species. Of these 146, 105 gave us enough genetic data to generate indices of population structure. So we used various indices and quantified phylogeographic groupings. And this is the data that visually, when we first looked at it, we realized something unique was occurring within our data. So here's just one species, Laodes oceanica. And what I'm highlighting here is that there are two clear genetic clades, and those two clades correspond to the two regions I'm showing you here. Now this pattern repeats and repeats and repeats across independent species. Laparoceras subnodosus, again, you can see two clear clades with quite a bit of sequence variation within them, structured into these two regions. The same for this as yet to be described species of Cardiophorus. And the species Acales globulipenis, and it goes on and on. And what is even more interesting is that in many cases, here I'm highlighting another clade in red, we find that these two orange and blue groups are genetically more different from each other than these two more distant groups here, okay, in Tenor. So we're finding that the red group here is clustered within this blue one, and the greatest separation is between these two groups and this orange one here. That's, quite, I would say, quite a surprising and astounding result at the time. So what we could do then was to summarize this pattern that we were seeing in a quantitative way to see, really, is it just our eyes pulling us towards, a few anomalous cases, or is there really something going on here? And we could undertake, using phylogeographic groupings, look at the relatedness of different sites in terms of shared phylogeographic groupings. And we find there's a clear and strong signal for regional level differentiation among beetles. Okay, here you can see we've got a hierarchical cluster analysis that clearly separates them. And the Permanova analysis generates a high coefficient of correlation, which is very significant. So that's looking at phylogeographic groupings, these groupings I just showed you in the previous slide. If we look at other less informative estimates of population structure like NST and GST, we still find this, strong signals for structure. And even just looking at individual haplotypes, we find a strong level of structure. But if we actually look at species, we don't. We find a very different pattern. What this tells us is that whatever's going on hasn't been going on long enough to drive species level signatures across this region. It's just patterns that's shared in common within species across the region. So probably quite recent factors driving what we can really witness as being incipient speciation across many species. Now, someone's asking a question. Oh, yeah. So what we did was we applied landscape resistance analyses to a number of landscape variables. Here we have them listed here, most of them related to water stress, to find out if these broad phylogeographic groupings or structurings relate in any way to landscape variables, or if it's more of a null or a distance-based pattern. And what we found is this phylogeographic groupings, the factors that clearly explain the division we see are elevation and mean annual precipitation. So, and that's the simple approach using mitochondrial data gives us, a clear understanding of a regional process that's occurring. And to really now focus in on that, we need to go down to individual species level. So what we did was just choose 8 species that show strong structuring, and as a comparison, 8 species that show no mitochondrial evidence for any structuring across this region. And we generated RAD sequence data. So if you like, it's like random SNP data across the genome for each species, generating thousands of SNPs from across the nuclear genome to compare with the mitochondrial pattern. Here's just the example for the 8 species that show strong mitochondrial structure. So here's the mitochondrial data presented as networks, and here you can see the divisions represented by these black bars between east and west. Here's the nuclear genomic data. Now, in every species, we find a coincident nuclear signature of an east-west structure. So this isn't really surprising, but it does help to reinforce the mitochondrial data. And beyond that, it enables us to look in more detail at what is happening at the individual species level. So here we have a principal component analysis that also reinforces that signature. And here we're generating indices of or ancestry coefficients that assign to each individual how much of its genome is ascribed to one or other ancestral population. Again, that defines all of these species as belonging to 2, or actually in one case, 3 ancestral populations. And what is super interesting here is that in 7 of those 8 species, we find signatures for admixture, and these admixed individuals occur in populations that are geographically proximate. So this nuclear data tells us not only is there regional structure, but it must be some kind of dynamic of isolation and secondary contact going on synchronously across multiple species. And when we undertook landscape resistance analyses for this, for each of these species, every single one, the individual relatedness was best explained by environmental variables, landscape variables associated with water stress, primarily elevation, temperature, and precipitation. In no models was geographic distance a significant explanation of individual relatedness. It's dominated by these landscape factors related to water stress. Well, as a comparison, here's the 8 species that show no mitochondrial signatures for structure. Basically, I can see a cat walking across some. Basically, signatures for panmixia. Right, just to summarize, for 5 of these species, the ones not grayed out, even though the mitochondrial signature tells us no structure, the nuclear variation tells us they show the exact same structuring into east-west regions. And in all species, we're finding signatures for admixture, we're finding signatures for admixture at these intermediate geographic locations. So this is the strong signatures we see in mitochondria are just simply amplified when we go to the individual species level. What we find is that there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 species that show exactly this structure here, east-west patterns with admixture here. Another species that has admixture shifted slightly to the east. And another species that shows 2 zones of admixture because there are 3 ancestral populations. And an additional species that shows structure like this here, but without any signatures for admixture because we didn't have samples here. So this is like massively, surprisingly repeated patterns across species. For all species, we generated estimations of when divergence started to occur using the mitochondrial data. And it tells us that the oldest event, the oldest species, its divergence is estimated to have occurred about 4.6 million years ago. And in the youngest species, about 100,000 years ago. And in fact, most species show divergences initiating closer to the present rather than this extreme age of 4.6 million years. So the obvious question is like, what's driving this dynamic? Now we have to bear in mind that these are species that are adapted to cloud forest habitat. They're species that are basically adapted to quite wet and relatively cooler environments compared to the rest of the Canary Islands. The two variables that interact to generate this are firstly a topographic deformation along this dorsal ridge due to a massive landslide that occurred 4.5 million years ago. So when this landslide occurred, which was about 15 square kilometers of the northern flank of the island disappeared into the ocean, it generated a depression here, an elevational depression of only about 130 meters, but it's the lowest point of elevation along the peninsula. Also, in this last 4 million years, we've had increasing climate oscillations due to glacial and interglacial periods in the primarily in the last, principally in the last 2.5 million years, where climate has warmed and cooled over short, relatively short intervals over this time period here, which in itself is going to have massive implications for cloud forest, because cloud forest is obviously directly related to global climate conditions and the cloud layer itself. That cloud layer, I'm just showing a small movie here, moves a lot in terms of its elevational limits on daily timescales, yearly and millennial timescales. So what we're seeing here is just how that cloud forest shifts its upper elevational limits over the course of one day. Okay, basically because temperature is changing, and that's one of the big, if you like, correlates of the elevational limits of cloud forest. So looking at this over more geological timescales, what's happening is as climate warms in interglacials and cools during glacials, the lower elevation limits of that cloud forest are going to be moved either upslope or downslope. Now, the traditional thinking, let me just, whoopsie. The traditional thinking is that what will happen is that as climate warms, cloud forests will be pushed upslope, both lower and upper elevational limits. And that intuitively makes sense. So what we'd expect now is that we should be at a period where cloud forest limits are at their highest and regional isolation at higher elevations is at its highest. But there's an alternative model that's gained some support, from different regions globally based on complementary data sets that suggest quite the opposite, that as climate warms, the lower limits of a cloud forest will be pushed downslope. Now that seems completely counterintuitive, I know, but there is good reasons to expect that might happen. And so we wanted to basically test which of these two models is occurring while synchronously understanding the dynamic occurring across all of our different species. So to do this, we went even on a more fine scale and shows one species sampled in more detail using this nuclear genomic data. This species, Acales globulipenis. And just to give the orientation of how we're looking, these are the sampling points across that peninsula. What I'm going to do now is just summarize the results of the ancestry coefficient analysis. Where are individuals pure and where are they giving signatures for being admixed. So here are the ancestry coefficients for all sampled individuals. We're just going to look now at where the admixed individuals are found geographically. Well, these individuals belong to this population. These individuals belong to this one. They're all tightly clustered in this narrow area, which corresponds to this lower elevation limit. Settle. I think someone might have their microphone on. Okay, so we know that there's admixture has gone on, and it's tightly restricted to a narrow geographic region. To really understand the dynamic of cloud forests, we need to understand the nature of those admixed individuals. Are they old relict populations of admixture, or are they ongoing areas of hybridization? So here's a principal component analysis of that same data. Here are the pure blues from the west, the pure red individuals from the east. And what we're seeing here across is showing extreme variation across the first principal component are the admixed individuals. Now, this is exactly the kind of pattern you would expect for ongoing hybridization, with recombination generating novel genotypes and very divergent genotypes within the area of contact. And we can confirm this with simulating expectations for hybrid genotypes, which we've done, and we find that the hybrid individuals we have are consistent with F1, F2, and recent back crosses. So what we have here is an area where hybridization is ongoing. And we can extrapolate this to all of our species, which show exactly the same geographical patterns of admixture and purity. So what we really have here now is like a really super interesting evolutionary scenario where we've got lots of coincident hybridization occurring across species, and that's something we now want to exploit. But in terms of understanding how cloud forests are responding to global climate change, this gives us clear supporting evidence for this alternative model, that when, I'm showing this here, that when climates are cool during glacial periods, cloud forests, the lower elevation limits of cloud forests are higher, and they remain like that during these glacial periods. And as climate warms and enters into interglacials, as is where we are now, those lower limits are pushed downslope, creating habitat continuity and opportunity for past isolated populations to come back into contact, which is exactly what we're seeing. And that dynamic is something that occurs repeatedly as climate oscillates. So what we're seeing is the most recent period of secondary contact, with many species showing evidence for repeated contact over, in some cases, several millions of years through this repeated dynamic. In the last 2.5, actually I can't, let me think. 1, 2, 3, 4, 5. Yeah, I think in the last million years there's probably been 7 oscillations of this night. So 7 events of isolation and contact. So it's super interesting from that point of view. And actually, that when we looked into this deeper, there is actually a climate model for the Canary Islands that was generated, which predicted exactly the same thing based on climate station data. That the climatically suitable range for cloud forests is likely to be extended downwards in a warmer world, and that's completely consistent. Well, that is completely consistent with this earlier model based on climate station data. Right. Now I want to move on and talk about how we can not improve upon this, but what next steps are, and finally finishing with some mention of dark taxa. Now, even though we created a protocol that is quite, in many ways, efficient, it still requires a lot of labor to invest in. We have to identify all individuals to a parataxonomic unit and sequence a subsample. So this involved sequencing thousands of individuals across these sites. What we're looking at now is ways that we can overcome that logistical hurdle. So rather than sequencing individuals individually using the Sanger technique. So one individual, one sequencing reaction, one sequence. We're now working with techniques where we can take all individuals from a site. So it would be 10 extractions for the 10 sampling sites, 10 sequencing reactions, and then generating sequence data that relates to the biodiversity or the species diversity of that original sample. Now, there's a lot of problems with this data. While it's in some ways very useful, there's also problems of sequencing and PCA error and other noise generated by nuclear copies, which has led to inflation of the estimation of species number and basically the need to collapse sequence variation to just identifying operational taxonomic units and having to really abandon any hope of using that intraspecific genetic variation. What we've been working on, when I say we, this is work led by Carmelo Andújar, and I think it's important that it's clear that he's been leading these bioinformatic approaches. We have come up with ways to remove or filter out the vast majority of that noise that has been problematic for people until now. And so in theory, permitting us to recover individual sequence data that's geographically referenced within species. So to test how well that works, we've compared metabarcoding data with Sanger data. So in this map, diamonds represent sites where we did our earlier Sanger sequencing, and these circles are new sites where we did metabarcode sequencing, whole communities, single sequencing reactions, thousands of sequences, and then bioinformatics to get individual sequences and then partition those into species units. So this is below is the original mitochondrial data. And here I'm showing you the mitochondrial data extracted from those bulk sample communities. Now the patterns are, they are just strongly congruent, even to the level of the population structure indices that we generate from that data. They are highly coincident. So now we have a mechanism to scale up, if you like, the generation of data. The only thing that you don't get from metabarcoding data is a good species ID because you've mushed everything up. But if you have a barcode reference library, then you can you can assign taxonomy after you've generated the species unit data. And that's what we've done here. We can, because we already generated barcode reference sequences for these target species, we can easily assign metabarcoding data back to those species. So just to sort of bring this presentation to a conclusion, I want to present some more work presented that's being led by Carmelo Andújar, where we're focusing on soil mesofauna. So we consider soil mesofauna as what we can call dark taxa. Dark taxa is a sort of a maybe not often well-defined term that just really refers to taxa for which we know there's a lot more diversity than we've actually described, but we can't really move forward with it because of the complexities of those taxonomic groups. Either it's cryptic diversity or just pretty large sample size, population densities. Those are the two variables. Small individuals with few informative morphological characters. So we're working to apply this whole organism community metabarcoding approach to really understand this kind of these kind of groups. And so we could be working on Diptera and Hymenoptera from malaise traps, but we're focusing on primarily mites and Collembola from soils, and Coleoptera because they're the next most abundant group in soil. So here I'm showing you 52 sites representing 4 different habitats where we've sampled soils. Now, after implementing our bioinformatic protocols, these are the species richness estimates per habitat. So we're estimating somewhere around 300 species using a traditional 3% threshold for defining species approximately 300 species per habitat. And if we just use a super conservative threshold of 15%, we're still finding, in the order of 200, an average of 220 estimated species per habitat type. And to give this some more context, in Tenerife, there are described and recorded 287 species of mite. We estimate from our data there are 434 species, and that's from a very, very small subsampling of soils across the island. So this is really, on one level, just giving us a massive insight into true species richness within these dark taxon groups. Now, beyond this, and I think this is the exciting part, is that because we're able to accurately extract haplotype level data with it at the species level within this metabarcoding output, we're getting information about habitat specificity, habitat generality, and whether things are showing signatures for specialization within habitats. So we have just got 3 as yet to be taxonomically identified lineages. I can't remember the details. This is work that's ongoing. It's not published yet. I think these are probably all mites, but there may be a collembola in here. So for example, this species, these are the different haplotypes recovered, and we're just color coding the habitats where they're found. So yellow is dry scrubland and red is thermophilus forest. So we can see that within this lineage, we've got clear differentiation within the lineage into the 2 habitat types. Again, in this species here, it's been sampled in laurel forest, pine forest, and thermophilus woodlands. And again, it's clearly structured by the non-random association of haplotypes with habitat types. And just so to not give the false impression that it's all like this, obviously many species are occurring in single habitats, and many are in more than one habitat and not structured by habitat. And here's a case here of a species that is occurring in laurel forest and pine forest, and there's no evidence for any within species differentiation into those 2 habitat types. So I think I've used up slightly more than the time I was supposed to have available to allow for questions. So now is a good time to, I think, draw my presentation to a close. Thank you all for your interest. If anyone wants to know anything more about this, I'm always happy to talk at any point. And I just want to acknowledge a few people who've been consistently inputting throughout these different dimensions of the project that we have that I presented today. Thank you very much.
Thank you, Brent. This is what happens when we have a smart scientist in a fantastic model island to understand evolution and speciation, and it's a fantastic. Congratulations. I now have some space. We have some space for questions. One moment. I can start probably by asking if you are also finding this kind of patterns in other Canary Islands, or at least if you have evidence of some natural history evidence or some already molecular evidence of similar phenomena operating in any of the other Canary Islands.
Good question. Good. In terms of the broad mitochondrial patterns, yeah, we're finding some interesting repeated patterns in La Gomera that we've yet to follow up on. But in terms of that last work I was presenting from Carmelo, that's analyses that are in progress now. So really we're focusing on this Tenerife paper as kind of it's just providing the framework. And then we have data for La Palma del Hierro and La Gomera, and from some other islands as well. And so we'll be looking at the same thing. And for example, we could compare across La Gomera and Tenerife, very old islands in terms of their laurel forest habitats, and La Palma del Hierro, which are very young, and see what sort of differences there might be associated with that. But I would say that we're going to see the same things everywhere, the same kind of structures, but just the proportional representation might change because of local differences between different islands.
Okay. Anyone with a question? João Gameiro wants to make a question.
Yep.
I don't. I think Sarah wants to make a question.
Ah, Sarah. Okay. Sorry, Sarah with the hand there. Okay.
It's okay. We look very much alike. It's normal that you confuse us. Thank you. So I'm really very much overwhelmed by the scale of this type of data. I'm definitely not a field biologist, so I'm always a bit scared with all this.
Yeah.
I have two very naive questions. The first is that, so you see a sort of a congruence in the spatial pattern, but do you also see a congruence in the temporal pattern? I mean, do they diverge? Do all these taxa diverge at a similar time or not? This is the first question. And the second one is also related to time, is do you see effects of seasonality, or have you looked at seasonality and how that affects, in particular, for example, I was interested in knowing the effects of seasonality on your soil diversity.
Yeah, I'll deal with both those questions. The first one, the timing of divergence, that's actually quite complicated because in many cases these things have undergone complex demographics of repeated divergence and contact. So trying to, the noise in that makes it very difficult to extract that. What we can say is that it seems most likely that they're coincident because there's no other sensible explanation for repeated patterns across multiple species with timescales that extend back, in some cases, 4 million years. What we're doing now is like, I think the real evidence would be if we find that all of those species are generating hybrids now, you know, so we have to resample many of those species, do the same level of detailed analysis, get more admixed individuals, because it could be possible that some of those admixed individuals are ancestral populations of admixture. And so, that's something we need to account for. But my feeling is that they're all going to be hybridizing now in these lower elevation regions because that cloud forest has been pushed down. Regarding,
but I'm sorry, but that doesn't tell you that they've all speciated at the same time, or that they've all diverged at the same time, right?
No, it doesn't.
Which would be really cool to
Yeah.
for your model of the.
Yeah. I think what's probably most likely, and this is just speculation until we get more data, is that different species started to enter into this dynamic at different times, and it could well be related just how strictly adapted to cloud forest conditions they are, because climate oscillations have got stronger towards the present. So you can imagine a species that's very, very tightly linked to these humidity conditions. When climate oscillations are fluctuating a little bit over time, it's enough to affect it. And others, more recently, the stronger oscillations we're seeing start to enter into the separation. The most recent interglacial was so strong that it's just started to create drying conditions that don't favor reproduction. Yeah.
So the seasonality.
Yeah, the seasonality. What was that question again, please?
So do you find evidence that this diversity also change along the year, right? Did you sample at different time points in the year, and do you see variation there?
Great question. We don't, we haven't sampled at different time points, but we're starting to do that now because we just don't know how much these things vary across the year. So Carmelo and Paula are doing this really detailed local scale study just to see how things vary across a small stretch of landscape, so lots of points in a small area, and then we'll go back through time and resample those points to just understand at a fine scale how it varies spatially, and over a temporal scale how it might vary. That's great stuff. Good question, and we're really interested to find out about that. Yeah.
Cool. Yeah. Thanks.
Okay, thank you.
Any other question? I don't see hands. No. Okay. And based on the data you have, we collected in Madeira and Azores, you have evidence of eventually similar processes occurring in Madeira Atlas, where there is complex orography. In the Azores will be more difficult.
More difficult,
but,
yeah, a good question. In Madeira, so we analyze those plots and generate the mitochondrial data, and I fully expected to find a structuring associated with the deep, I can't remember, what's it called? There's a deep valley that bisects the center of Madeira.
Ribeiro Frio.
Yeah, exactly. So there's, like, if you like, two clear regions bisected by a narrow, low elevation region in the middle. But no, we find nothing. So it could just be that I'm guessing that Madeira is slightly, it just has a very different climate to the Canaries, and it doesn't favor the drying conditions over that landscape that would be required. We might find some evidence, obviously, if we went and looked at individual species with rad sequencing, take into account that many species in the Anaga study that showed no structure with mitochondria showed it with the nuclear data, probably because they are just starting to get impacted by these events over more recent climatic oscillations. So it could be, I'm not discounting it, but if it is happening, it's on a more recent time scale than we're seeing the dynamic extending back to in Tenerife. And we need to look at the individual species level with nuclear data to go for that.
Then the last question, because time is flying, to Otávio.
Hi, Brent. Can I ask you about if you plan to go to other markers, nuclear markers, and try to find the reasons of that adaptive differentiation that seems that you are finding?
You mean for the soil mesofauna that we're finding? Yeah. That would for that. I mean, that's obviously something that would be interesting to do. I think the complications there are, for a start, we don't have a species ID for those things. Now what we have to go and do is generate a barcode reference library to say, that variation corresponds to this type here. And then working with these very small organisms kind of complicates genomics approaches. I would say that these are definite things that can be followed up on. But again, it's one of those situations of like so many lines of interest that, you know, some things just have to be left to the side for now.
And congratulations for your amazing talk. Okay.
Yeah, thanks. Yeah. I can't hear you, Paulo. I think you're on mute.
Okay. Thank you, Brent, for this presentation, to accept this invitation. In these difficult times where you are very busy, and enriching our conference. And I hope you can come next year to Azores to our C3C conference in Terceira. You are already invited. I hope at that time you can find also time, some space to visit us.
Yeah. If I can, I will. Don't you worry. And just I'd like to thank you for the invitation again. Thank everyone for listening in, and make an apology that I won't be able to stay and listen to people's presentations as my son has decided to get ill, and so I'm going to have to be sitting with him today. So hope the rest of the day goes well, and thanks again for listening in to my talk.
Thank you, Brent. Okay, thanks. Bye.
Bye-bye.